Hello and welcome to Learn A-Level Biology for Free with Miss Estrick. Today we're going to be going through the biochemical tests that you need to know for the biological molecules unit in A-Level Biology. You will probably find it helpful to have some paper nearby so you can make a note of the tests' positive results, and there's one question slotted in this time as well, for which I'll give you a reminder just to pause the video at that point. So the biochemical tests that you need to know are tests that are linked to looking for the presence of 3 different types of carbohydrates, proteins and lipids. And the carbohydrates, there's only one of the polysaccharides you need to know the test for, and that is for starch. The reducing sugars test you would have learned at GCSE, but we go into the details of why it's called a reducing sugar this time. And also a non-reducing sugars test, which will be new. So the test for starch, first of all, the reagent that you have to add is iodine. And in this case, I've got my sample of starch. Then you would add your iodine, which is a browny orange colour. If you have starch present, you get a bluey-black colour. So the reagent is iodine. Positive result is orange to blue-black colour formed. Test for reducing sugar. So this is the one you would have learnt at GCSE. So in this example, I'm going to be testing glucose, which is a reducing sugar, but also show you the result with just distilled water, so you can see what a negative result looks like. So I've added Benedict's, which is the bright blue reagent, to both samples, and you have to heat for this reaction to work. So the glucose test tube, you can see, is starting to change colour. Water remains blue. And that's what our results would be. So for a positive result, you go from blue to orange, for example, glucose. Negative result, it remains blue, and I used water as an example. And you can get other colours other than that bright orange, that brick orangey red colour I've got there. And the different colours would indicate the concentration of reducing sugar you have. So if you only had a green colour or greeny yellow, that indicates a low concentration of reducing sugar. The more orangey red it is, the higher the concentration of reducing sugar. Now you might have noticed in the beaker, as the reaction was occurring, that the colour changed at the top of my solution first. And I've taken a shot from the video here where we can see that bright orange colour starting to form at the top first, and it's yellowy green and almost still blue down here at the bottom of the test tube. So this is the point where, just to pause to have a think, why is it that the reaction is occurring first at the top of my solution? So this links to physics actually, and it's to do with the idea of convection currents. So the hotter particles in the solution are going to be rising, and therefore the hottest point in the solution in the test tube is at the top. So the molecules will have the most kinetic energy at this point in the test tube. Therefore, more successful collisions, and the reaction will be happening faster, and therefore colour change is happening first at the top. So next, the non-reducing sugar test. The only non-reducing sugar that you learn about is sucrose. So I'm going to test a sample of sucrose. First thing you have to do though is confirm it isn't a reducing sugar. So I'm going to do the Benedict's test first, which we can see, leaving it in the hot water with the Benedict's for a few minutes, and we've got a negative result. So I'm adding sucrose again to now go on to the next step, and this is acid hydrolysis. So I've added acid to my sample of sucrose, and this time you do have to get it to boiling. So I've got my thermometer in the test tube to make sure the acid does get up to 100 degrees C, so it's boiling, and it has to be boiling for about 2 minutes for acid hydrolysis to happen. And what we mean by that is we're going to hydrolyse the glycosidic bonds, so the sucrose is going to be converted into glucose and fructose. So that's our next stage in the video. We have the boiling acid for 2 minutes. I am then going to move it into a beaker of cold water to cool it down for safety reasons. And once it's cool, add an alkali. Then we can add Benedict's and heat again, and we'll get a colour change. And you'll start to see again at the top it's going a red colour until eventually the whole solution we've now got this brick red precipitate. So the brick red solid is formed at the bottom. So that's our positive result. When we did retest with Benedict's in heat, the Benedict's went from blue to this really dark brick red colour to confirm we do have a reducing sugar. So just to summarise then what we mean by the reducing versus non-reducing sugars, the reducing sugars are all of the monosaccharides that you learn, so glucose, fructose and galactose, and 2 of the disaccharides, lactose and maltose. So the only non-reducing sugar that you learn about is sucrose. And the reason we call them reducing versus non-reducing is these 5 sugars are able to reduce copper sulfate, which is the blue chemical in Benedict's reagent, and they reduce it to copper oxide. And copper oxide is a rusty brick red colour. So that's why it does convert from that blue to the brick red colour. Sucrose, which is our non-reducing sugar, isn't able to reduce copper sulfate. And that's because the group within these sugars that does the reducing is actually used up in the glycosidic bond in sucrose. So it's not going to reduce copper sulfate to copper oxide. Hence the name, it's a non-reducing sugar. The reason it does change colour though eventually is we hydrolyzed sucrose. So we boiled it in acid, and in doing that, that breaks the glycosidic bond between the monomers glucose and fructose. So we now end up with those 2 separate monomers. And when we then do the Benedict's test again, we do get that brick red colour. And often you'll find it is a very, very dark brick red colour like we had, because you now have 2 monomers in there, glucose and fructose. Whereas when we were just testing for glucose in the previous experiment, we just had 1 sugar. So we had more sugars present, that's why it was that brick red colour. So next test is for proteins, and you would add biuret, which is another bright blue solution. I've just emphasised the pronunciation here, so biuret, because often students call it biuret, which is incorrect. That is a piece of equipment that you use in chemistry. So biuret is the name of this reagent, and I've got albumin as the protein that I'm going to be using. So I'm adding my sample of protein, adding the bright blue biuret, and a positive result is it turns a purpley lilac colour. So the last test is the test for lipids, and I'm going to use oil as my lipid. And step 1 is you have to dissolve the lipid in ethanol. So to dissolve it, bung on top, shake it up, and it is dissolved. The next stage is you add distilled water and shake it again. Positive result is it's white in colour, and it's an emulsion. So you would have to state both of those parts, the colour and the fact it's an emulsion. There's no precipitate, there's no solid that has been made, it is just an emulsion. So in summary then, the test for monosaccharides and the disaccharides, with the exception of sucrose, is the Benedict's test. The test for sucrose, which is our non-reducing sugar, would be after a negative Benedict's test, you would then go on to do acid hydrolysis, which is boiling an acid, neutralise with an alkaline, and then heat with Benedict's again and observe the colour change. The test for starch is iodine test, lipids is the emulsion, and proteins is the biuret test. Now that is just a brief summary. In an exam, you would always have to state which chemical you're adding, which is the reagent, the conditions, so that could be whether you have to heat it, whether you have to boil it, do you have to mix to dissolve, and lastly the colour change to indicate the presence. So state the starting colour and the positive colour result. And that's what we're going to finish on, just summarising all of the positive test results. So lipids was the white emulsion. Proteins is this purpley lilac colour. Starch, you get blue-black. And reducing sugars, you can get any colour from the green, yellow, orange, brick red. The more dark red it is, the higher the concentration of reducing sugar. So we've got a dark orange in this case. So that's it for the biochemical tests. You need to know the method and the positive test result. So that's it for our lesson today with Learn A-Level Biology for Free with Miss Estrick. You can click on the link to go back to any of the biological molecules which were mentioned in this test, just to learn the theory on them if you wish.